I'm commanding the microphone, folks. All right. Well, it is so wonderful to see all of you here today to support Gareth in the culmination of what is a tremendous amount of work. I am Bonnie Nagel. I am the faculty member that has had the great privilege of working with Gareth for the past lot of years, both as a research assistant in my lab and then as a graduate student. I'm thrilled to get to introduce him to you today. I could spend days, but it would take me months to do all the digging to get all the dirt, and I didn't have months to do that. So you're gonna get a snippet. So what I'm gonna say about Gareth is that he has been really fun to get to know all of these years. And I'm gonna tell you just a few things about him. Like I said, I could speak for days about how awesome of a human he is, but I'm just gonna share a few things. So he loves a good donut. And occasionally he loves to eat what might be nearly a dozen raw eggs and then come to a meeting with such bad intestinal distress that he might die and tells us about it. Gareth also is a fierce advocate of having compassion for both himself, it's growing, and others. And he makes a hell of a fantastic chocolate chip cookie to help lure people into his compassionate efforts. I have had those cookies. They are really, really good. Occasionally, he just needs a nap. And that nap could be wherever he might be, including in our lab. Yes, he's just sleeping. I asked Amanda, is he just sleeping? Yeah, he's just sleeping. It might have been after the eggs. I don't know. Hard to say what he ate that day that contributed to it. He's also very serious about working out. Now, I know there are people here probably who work out with Gareth, and I wish I had your cell phone numbers so that I could have gotten some great pictures of this. But I have seen the flex of the chin, of the calves. I've seen the flex before, so I know Gareth takes this very seriously. So much that he wants to compete in this arena, and may. Ooh, and now I'm realizing as I put this together, I didn't put anything about his musical inclinations in this, but I could have played some really amazing music for you all as well. He embraces the moment. This is hands down my favorite picture of Gareth. This is at a lab camping trip many, many years ago. And yes, he is in a freezing cold Oregon River in this picture, just striking a mermaid pose and just being himself. And of course, as we'll see in a moment, Gareth is truly a brilliant scientist and an excellent presenter. And I am just so proud to have you all get to experience the journey that he's been on with his dissertation. Gareth, I'm very, very proud of you, and I'm looking forward to your talk. I can't get this to turn on. Go ahead and get yours. Yeah, I can't.
That's very good.
Hold on, I'm moving out of your way so you can plug in. I'm gonna shut this down while you plug in.
Thank you, Bonnie. Some of those things were true. Most of them were true. I do nap a lot, or try to. I like to lay down more than nap, I would say. All right. I think we are ready to get started. I wanted to double check. Is Jay on the able to make it? Jayashri. Yeah, sure. But I think we could get started. We could get started, right? Yeah. All right, so this is my dissertation. It's pretty wild to say, dissertation defense. And we're gonna talk about a lot of stuff today. We're gonna talk about AI and structural MRI. I should probably stay near the microphone. and specifically trying to use it to improve our ability to predict these aspects of self-regulatory behavior and mental health. I want this to be approachable. I want to stay on the slide that we were on. But I want this to be approachable. It's a complex topic, but so I'm going to spend a lot of time on background today. And I think when we think about these mental and behavioral health disorders in childhood and adolescence and look at sort of the incidence of these disorders in this population, it becomes clear why we might want to study them further. We probably know somebody young in this room that is struggling with a mental or behavioral health disorder. But our mental health, it's very difficult to study because it's such a complex aggregate of all of these different symptom domains. We have our social relationships, we have aspects of environment, and then this very real piece of biology, and with it, the brain. So the brain is not the picture of mental health, but it is a piece of that picture. So this is what we're focusing on today. But because it is so complicated, we have a lot of people in different domains working on this problem. I want to take a second to say where I fit into this because I forget a lot. But I am very heavily embedded in the computer science realm, as well as, no, that's it, and the informatics realm. I'm in a computational biology program. I'm not a neuroscientist. I am not a clinical psychologist, but I do work in these domains, but my interest is within the data, and specifically trying to model how the brain either contributes to or protects us from these mental health disorders. This is my overarching objective as a researcher. And on the sort of typical timeline of these events, right, we define a problem, we start collecting data, and then we move towards clinical implementation of that model to improve the life of the population that we're studying. We today are going to focus very heavily on this aspect in the middle. Because I'm an informatics data person, we're going to focus on how we deal with the data, how we model the data, how we validate the data. Hey, Francis. And then moving forward with that, and specifically focusing on this aspect of model development, and specifically with these new tools like deep learning and AI. So what are we going to talk about today? Poster pitch. Psychiatry is very hard. It is very hard. We'll talk about why imaging is hard. We'll talk about some academic and not personal failures. We'll talk about what we know. We'll have some, relief, collective relief, and talk about some results, as well as talk about some conclusions as well. But we start with psychiatry is hard, because psychiatry is hard. It's very different than other types of diagnostic disorders, right? Where somebody comes into the clinic and they get this sort of non-subjective physiological measurement that we readily use to bin them into diagnostic categories. But in mental health, we have these sort of subjective measures that are endorsed either by the participant or the parent of the participant and aggregate them onto these disorders. So if sweet, poor Brendan Fraser comes into the clinic and he is having a hard time because Encino Man 3D went straight to DVD, and so he is having issues concentrating, he has fatigue and low energy, he is sad, and he's having sleep disturbances. If we want to give Brendan Fraser a psychiatric disorder, we might just count these symptoms. So we count symptoms that load onto specific disorders, and maybe the diagnostic threshold for major depressive disorder is 4 symptoms. So Brendan Fraser is struggling with depression. But there are a lot of problems from a research perspective when looking at these sort of binary diagnostic categories. We have a lot of heterogeneity within these disorders. So 2 people with depression could present very differently. There's also comorbidity. So people have oftentimes multiple mental health disorders. There's a lot of shared overlap in some of the symptom presentation. Something like difficulty concentrating could really be a hallmark of any one of these psychiatric disorders. And we have these sort of discrete outcomes that are tough statistically. So somebody that has 0 symptoms of depression might look very differently from somebody, both arms in this one, than somebody that has 1 symptom below the diagnostic threshold. So rather than looking at binary diagnoses today, we're going to focus on these broader domains of externalizing and internalizing psychopathology. And these become very attractive targets for research purposes because they account for much of the shared variance in symptom presentation. Disorders like major depressive disorder or panic disorder, they're going to present sort of similarly symptom-wise. And they are these nice continuous variables that we can model along this spectrum. But we still have this issue because we want to use the brain to model mental health. And there's a lot happening in between brain and mental health. There's not a depression circuit in the brain. We already know that mental health is comprised of these symptoms. There are symptoms that sort of instantiate these disorders. And within each of these symptoms, we could decompose it even further into specific aspects when we're designing like task fMRI studies. So things like how we anticipate reward, or how satiated we feel when we receive reward, or how we appraise threat or process fear, or if we're having difficulty concentrating, it could be issues with memory systems or our ability to sustain attention. So because it's so hard to model mental health, and the goal of this project is to try and evaluate these emerging modeling strategies, specifically deep learning, we also are going to look at sort of an intermediate target in the form of these self-regulatory behaviors, sometimes called aspects of cognitive control or executive function. These are a very important set of processes that are tied more replicably to these neurobiological correlates. And deficits within these processes are associated with every form of psychopathology. So we have this nice sort of possibly easier to predict target than something as complex as psychopathology. And specifically today, we're going to focus on working memory and externalizing psychopathology, which has a pretty robust body of literature implicating deficits in working memory and externalizing psychopathology. And we're going to do that using data from the Adolescent Brain and Cognitive Development Study, which is really this landmark study of nearly 12,000 children, aged 9 and 10 at baseline, recruited all across the United States. And it's hard to understate how significant this study is in terms of the population when we have just a ton of data on these individuals, which is pretty fantastic. We have behavioral measures, genomic information, and then the neuroimaging data, which brings us to imaging is also hard. So together they are very hard. And we're gonna do the very trite, like, what is an image? Webster's dictionary defines. We're not doing that. But I am gonna show an image of me as a baby with my mom, who I'm looking at right now. And I'm making a fun smirk, which is really nice. And what I want us to remember is that an image is just a matrix of numbers. We have to think about how the computers deal with this data. So it is some number of pixels wide. It is some number of pixels tall. If we're looking at a color image like we are here, we have red, green, and blue channels. We have this third dimension. And so if I take this image and I zoom in on a specific piece, like my sweet little double baby chin, and I pick a specific pixel, there are going to be 3 numbers representing colors of the colors, I don't know, just the colors of that region of the image, red, green, and blue. They're just numbers. If I make this image grayscale, I remove that third dimension, and now I just have a single value representing overall intensity. So I want us to focus on this because it's hard to use imaging data to model these targets. So if we have a task where I have all of these handwritten images, which are these numbers, and I want the computer to classify what number they are, we sort of immediately run into these problems because most traditional algorithms, for example, linear regression does not want 2-dimensional data. God forbid 3-dimensional data, which is where we're working with. Amanda's laughing. And so one of the things that I could do with this is I could flatten this data. But if I do this, if I choose to flatten my data, any transformation I apply to my data is going to alter it in some format. So if I show you these other 2 flattened images, you cannot tell me that that is a 0 or an 8. I would call you a liar because there is context embedded in that native dimensionality, in that 2-dimensional space that we lose when we flatten the image. So it's really tough. We're sort of limited in how we can use this data. The other way that we often extract bits of information from this image is we could measure some high-level properties within the image. So in this case, I could extract the volume of the non-zero pixels, these white pixels, or I could measure the longest linear contiguous segment of non-zero pixels. I could get at these high-level properties of aspects of the image. And this is sort of exactly what we do with the structural MRI. So we would take a piece of software called FreeSurfer, and we would segment the brain into different regions based on this anatomical atlas. And with that, we would get these measurements, things like volume, thickness, intensity, surface area of these structures. And we use these values moving forward for prediction. But there are issues with this, right? We've talked about loss of information. So if I ask Pete, are these images the same? And you would say, no. He would say no. Put him on the spot. They're not the same. And if I asked him why, it might be very hard to articulate this because there are very low-level properties of cortical folding that tell us that these are not the same images. This is a slice of the superior frontal cortex of these two different subjects. And maybe you could point to specific regions. You could look here and say, well, like the folding in that region looks a little bit different, or maybe some of the folding here doesn't look exactly the same between these two subjects. But again, when we apply and get these high-level properties from the image, we lose these very complicated low-level patterns within the data. And specifically, this is the wow moment, where we look at the manually extracted features, things like surface area, volume, or average thickness, we find that these subjects are almost perfectly correlated. We know that they're not the same, but when we get those high-level bits of information from the data, we lose that low-level nuance. So this brings us to the primary hypothesis. It took a while to get there, but the primary hypothesis being that there's just information in the data that we don't know about or we can't really readily get at ourselves. And then on the heels of that, equally important is this aspect that this information has predictive utility. It's something to know that we're not getting all of the data, and another thing to say we can get that data and show that that data is useful in subsequent prediction. But again, this is hard. This is a hard problem. There's a reason we look at these high-level features. But what we're going to do is use, again, deep learning in the title of the talk, to evaluate if we can't get at some of this information to help us make these predictions. So deep learning is a subset of machine learning, which is a subset of AI, which is computers learning patterns and data. And you specifically, everybody, uses deep learning every day, whether you know it or not. Your iPhone camera, that cool thing where it makes you look old, voice assistants, autocomplete, recommendation systems, and there are just some tasks that are too complex for these traditional algorithms. For example, autonomous vehicles. You could never have a linear model take in video data in real time and do real-time object detection and make decisions about where to drive. That's a very difficult task that requires the flexibility of something like deep learning. Yeah, that was the end of that. And then we're going to evaluate that today in the case of healthcare diagnostics. And there's a lot of hype around AI. I will be the first to say that there is hype, but it can do some really cool things. I think many of us in this room have used something like ChatGPT, and generative AI is really interesting. So I could ask it something, right? Generate me an image of Tommy Pickles snatching 125 kilograms, and it would try, and it generates this fun image. He's got, you know, he's on a platform, he's got a belt on, he's got a diaper that's very full, and the barbell is, you know, it's going through his head, but it's pretty good, right? It's pretty good for generative AI. And I could ask it to segment this data, apply segmentation, and it would generate one of the scariest images that I've seen in a while, and you can buy this on my Etsy. This is not my Etsy, so maybe don't go to it. But it's fun. It's a fun idea. I can make an Etsy. But the point being that deep learning does have a lot of really cool aspects. It is extremely powerful. It can model these complex data structures, things like video data or imaging data or graph objects. It is highly customizable, meaning that I could structure it in whatever way I want. I could extract bits of information. We'll get into it. I'm getting ahead of myself. But there are caveats to each of these pros. So it is enormously powerful, but because it's powerful, it requires a lot of computational resources, something that we don't always have available. It can handle these complex data structures, but it needs a lot of data to learn patterns in really high dimensional, difficult to work with spaces. It is customizable, but I can speak to this aspect of tuning purgatory, which Nate knows about, which is getting sort of stuck, making small tweaks to the network where you're trying to improve performance and you stay like a year there and you start to spiral, and it's easy to stay there. But it is cool because it can do these cool things. There's a lot of flexibility, meaning I could take my structural MRI data, I could feed it into my network in such a way that I extract those low-level properties from the image. And then once I get those low-level features out, I can combine it later with clinical data, and I could combine it with covariates later in the model. And this is really cool. This isn't flexibility that we often have in some other modeling strategies. And I keep saying this piece of extract information from the image, and I want to take a second to talk about this because I think this is where I lose a lot of people. And it does so using these convolutional layers. They are convolutional neural networks that we're evaluating today. And the convolution operation, it is math, but it's really intuitive, right? In signal processing in a one-dimensional space, this is how we would represent it. Two-dimensional case, we would make some modifications, and same for 3D. And I kid because I know these functions look very intimidating, but intuitively they are pretty straightforward. In the case of that 2D image, I'm going to take one image, slide it over another image, and then make a third image. And what does this look like? So in the case of, again, a single slice of the superior frontal cortex of a given subject, I can create these two other images. And again, images, matrices, the colors just represent negative or positive values. And what I can do is I can slide those kernels over the image, combining them to generate new images. And so what does that look like? If I take these two different kernels, slide them over my image, combining them at each section, I can get at other low-level properties within the image, these sort of horizontal boundaries or vertical boundaries. So in the case of the kernel on the top, I'm getting at horizontal borders of brain, no brain, or gray matter, white matter, or that's it. But this is very flexible. We can do a lot of things with this. We can do this in abstraction. I could keep going. We could get the vertical lines of the vertical lines or the horizontal lines of the horizontal lines. And that sounds like a lot, but it's very powerful and flexible. And we do this all within the context of the neural network. So it will, is that the way? No, it does it in the context of the neural network. What I want to specify here is that in this example, I am specifying the weights in these kernels. Applying kernels to imaging data isn't inherently novel. But in the case of the neural network, what makes it deep learning is that it is learning the values in these matrices in order to get information from that data to best predict whatever task I've given it, whatever that is. So in a little more understandable case, if we're trying to predict a picture of Carl, it might learn aspects of the ears or low-level patterns of the eye or the nose, and it's going to aggregate those low-level features hierarchically to generate these more complex structures within the data, like an entire eye or an entire nose. So this is how we're learning these patterns, and it's very powerful. I said it's very powerful a lot. So I'm gonna take a sip, and then we hit the ground running, right? We are bright-eyed and bushy, and we say, okay, multimodal data. This is sort of like the holy grail of neuroimaging. We don't know the most about structure and function together. So we say, I'm going to build a neural network that is going to extract these low-level representations of function and then structure function together. And it's going to be super great. And it just totally didn't work. Meaning that I could not predict either working memory or externalizing psychopathology better than random chance, which is where we get to just a couple years of my life. And we're going to talk about this, and we're going to talk about why. And I like this piece because it's important in informing what we eventually tried that did work. So we admit, I like how it's like me admit that I was a little overzealous. I had to convince my deck to let me do these very complex networks. Sure they'll work. I'm sure they'll work. And they didn't. So we start to say, okay, maybe we were a little overzealous. I was a little overzealous. It's very difficult to take high dimensional functional and structural data in the same network. These data are enormous. The structural data has 7 million numbers within it, and learn these low dimensional patterns. There are a lot of patterns of cortical folding within this structure. It's moving slow. So we're going to split it up and then sort of more circumscribe individual tasks for these networks. And this wasn't working. So we start to panic, a little bit more, and we say, okay, I'm gonna simplify the networks. I'm gonna add layers. I'm gonna remove layers. I'm gonna change operations. Nate's talking about graph neural networks, and we don't listen to Nate, except we do. I do appreciate it. And we keep moving forward, and this wasn't working either. So then we start to really dig deep and say, okay, well, maybe the predictions aren't meaningless. Maybe they're like right below meaningful. And if I aggregate information from many sort of right below meaningful networks together in these ensembles, and again, Nate starts talking about uncertainty quantification, and it's a very cool idea. And I do appreciate all of Nate's suggestions. And it's not working. These ensembles are not working. Yep. Yeah. So then we start to say, okay, let's again go back to the wheel and say, we're going to focus on structure, right? The functional data is really cool, but it has very low temporal signal to noise. It's very noisy, and it has a lot of motion, other issues with it, and it has a lot of subjects that don't pass quality control. So we have to exclude this large portion of our sample that don't have usable functional and structural data together. So we say, we're going to focus on the structural data, and I'm going to include these additional 2,700 subjects that we weren't able to include. And I am going to include something I know about the brain. I am going to break this down and simplify the task. And there's two ways that we can do that in the case of these neural networks. I can include information that I know, or I can reduce the size and complexity of the input. So I already have these segmentation masks. We've done the segmentation using FreeSurfer to get at those high-level features. So we have these sort of like relatively, more anatomically distinct regions, and they're a lot smaller. So instead of focusing on this entire brain in a single model, we split it up into different models for different structures, such that we are now training many networks, but our input is a lot smaller and it's more circumscribed. And it also just totally didn't work. And so we start doing things. We start lowering the resolution, and we start applying these minimum bounding cubes, and we just fully panic at this point. And so we start asking questions. And this is just for narrative, because we've been asking questions the whole time, right? But let's pretend that just now, it's like, why isn't it working? And so the most obvious question becomes that there's just no relationship between the brain and my outcome, which would be devastating. But we know this isn't true, right? We've been running models this entire time. We know that these high-level bits of information, like volume and thickness, predict both working memory and externalizing psychopathology. These high-level bits of information do. The deep neural networks are not working. We also have this pretty robust body of literature that indicates that we should be able to do this as well. So we start to go back to the way that we're modeling the data. Again, very complicated data, very noisy, very big. My outcome is very tricky to model, but we are stubborn, and we still believe that there's information that we're just not capturing that we can get out that might be important. So again, what is and what is not working? In the case of what is working, extract, then model. When I'm extracting those high-level properties from the image, I'm not thinking about my outcome at all. I'm just getting aspects of structure or morphology, the same thing, and intensity out of that data, and then subsequently using that information to model my outcome. What is not working are these supervised neural networks, where I'm trying to put in my very raw, very large data to directly predict my outcome. Which again, it's hard to understate how big and difficult to work with this data is, both just computationally and I guess just computationally. And our outcome is really tricky, right? It's really difficult to model psychopathology, less so for working memory, but still difficult. But we still believe that these operations in these neural networks are powerful, just maybe not directly in the context of our outcome. Maybe we want to do this in an unsupervised setting, which means that we're going to use the neural network to extract information out, those low-level features. We're just going to get them out. We're not even going to think about our outcome right now. And we're going to do this using this architecture called the autoencoder. And the goal of the autoencoder is it's pretty cool. We have this encoder-decoder structure where we take our very complicated three-dimensional object, put it into these layers, and these layers extract these low-level bits of information, compress them into this very small representation of numbers in a one-dimensional space, and then use that space to try and, as accurately as possible, reconstruct that original image. With the sort of theoretical belief being If I can use that very small space to accurately reconstruct my data, then that small space must capture very important bits of morphology and intensity of my data. So we try this and we move forward and we say, okay, I am going to evaluate, again, the free surface statistics, things like volume, thickness. These are, again, like the most high-level properties of the image, but sort of the most common way that we look at this data. We are also going to use another modeling strategy called PCA. PCA is not deep learning. It's actually pretty straightforward. It is a linear method of reducing the dimensionality of our data in order to best retain variance or variability within that data. So we're going to compress it into the smaller space, but we're not using any of the embellishments of deep learning yet. And there's another piece. And we have to flatten the data because it's a traditional algorithm. It does not want that three-dimensional format. So we flatten this very large data, which feels comical when I think about it, but we flatten it into this very long one-dimensional vector. So we'll find out if that 3D structure is important in the subsequent prediction. And then we move forward with the autoencoder, still focusing on this aspect of deep learning and AI. And the goal, again, of the autoencoder is take that complex three-dimensional structure, compress into a very small space, and then reconstruct. So we have these three sets of features. Again, they all have different objectives. They represent different characteristics of the image in that the objective of generating them is different. So how well do these aspects predict working memory? Well, you need to hold on because we need to evaluate that these things are actually doing what we think they're doing. If the autoencoder is not able to reconstruct the original image, those features are going to be entirely meaningless. But it can. It can do it very, very well. It can take this enormous 500,000 numbers, this region of the cortex here, and compress it into a space of 20 numbers, 500,000 to 20. And almost perfectly use that very small set of features to perfectly, not perfectly, but very closely reconstruct these very complex patterns of cortical folding with minimal error. This was pretty, I thought this was pretty cool in itself. PCA, however, is not very good at reconstruction. We can see that. It's not really getting sort of these more nuanced patterns of cortical folding. But what we have to remember with PCA is this isn't the goal of PCA. The goal of PCA is not reconstruction. All of these algorithms have to be considered in the context of their objective functions. What are they trying to do? And in the case of PCA, our goal is retain variance. And we can do this pretty well. We can retain 20% of the variance and cut our feature size from 500,000 to 20. So we lose a good chunk, but in the case of modeling, we are always battling dimensionality or size of our input and just how complex that is, I guess. That's the only bit there. So we can compress it very significantly and retain a substantial portion of that variance. So now how well can we predict working memory? Well, we knew that we could do this using some of those high-level statistics. We see, again, we see some orange in there and some red. Here on the right, we have the performance, which is correlation between actual and predicted. So red is better. And so we find a couple regions, the middle temporal gyrus, precentral gyrus. Can't point to any of these. Some of the redder regions. And then what we see when we start using information from PCA is a lot more red, a lot more orange. We get better performance using information from the data that we weren't using before. One of the things that I really want to highlight is within these subcortical structures. So they were not predictive at all. Gray is that they were not better than random chance. And in the case of the free surface stats, the conclusion that we would have come to when running these models, a very common way that we would have done it in other analyses, we would have said there is no relationship between morphology of these subcortical structures and working memory. These features of, or these regions of, emotion and reward, they do not predict working memory. It's just, wait, no, this is not true. It's just that the information that we had available to us did not capture the bits that we wanted to predict working memory. When we go back to the data and extract more information, more nuanced aspects of variability, we can now use these structures to predict working memory. The autoencoder. It's hard to say. It's hard to say when, there's a lot going on in these plots, but we see maybe a little more red, but the deep learning is working. It is working now. So we, do a little clap that these models are actually working. And they can do pretty well. And what we find is when we use all of these bits of information together, so information from the free surface stats, the PCA components, and the autoencoder together, we in some cases get improved performance. And again, hard to tell with this big figure, so let's focus on the top 3 predictive structures. We have the middle temporal gyrus related to language, semantic memory, visual perception, aspects of association between morphology in disease states, the inferior temporal gyrus, and then the precentral gyrus. These are the top 3 structures that predict working memory. And one of the very interesting, okay, so let's go through the colors. It's sort of hard to see, but on the left is red, which is free surface stats. The yellow is features from the autoencoder, deep learning. Green is these features from PCA, and then blue on the far right is all of these bits of information together. So what we see right away in the top performing structure, that of the middle temporal gyrus, is a performance that is better than using all of the information from the rest of the brain, those high-level bits of information. So with a correlation of 0.25, the best we could obtain using all of the features from FreeSurfer of all regions was around 0.24. And so we can do this using information from this single structure in the brain, which is very interesting. We also see that this yellow performs better than red, which is that deep learning in this case is outperforming that of the FreeSurfer stats. This is not the case in the inferior temporal gyrus. We see that the red actually does the best of the individual modeling strategies, but when we incorporate information from all of these 3 sets of features together, we get the best performance. And then subsequently within the precentral gyrus, we find sort of equivalent and superior performance of both the deep neural network and PCA. So immediately we've answered that question of, is there information from the images that we're missing? Yes. Does it help us predict our outcome? Yes. Regionally, it does. Including these bits of information improves our performance. This becomes even more notable when we look at some larger structures, like all of the cerebral white matter. This again is a very big piece of information, and the information we have from the FreeSurfer stats is very, very limited. It's not really the goal of this modality, but if we're looking at all of the cerebral white matter and the information we have from FreeSurfer, we would say not better than random chance. But when we put this structure into the deep neural network, we find that this information becomes significantly predictive. Or if we include information from PCA or all of these features together, we also improve our performance. PCA doing very well. One of the quick caveats that I want to mention here is, again, thinking about the data and what this means sort of conceptually. So I have this piece of, is it actually the morphology of the cerebral white matter? Yes and no, right? The cerebral white matter borders the cerebral cortex. So we also include a lot of information about surrounding cortical structures. When we're getting this information spatially, we need to consider other information included in that structure. Just a caveat, not that it's not important, but remembering what other information could be encoded within the structure. The next piece becomes aggregation, right? If we can predict working memory this well using a single structure, how well can we do it using information from the entire brain? So the first way that we're going to do this is by using the entire brain. We're going to take the entire brain, dump it into a single autoencoder, single deep neural network, or a single PCA model, which again, pretty comical to take 7 million pixels, flatten them into a one-dimensional vector and put them into this very, very large model. The other method we're going to try and evaluate is that of using information from all of these individual structures together, both from the PCA models and the autoencoder. So we're including all of these bits of information, concatenating them, and then using this information to make our prediction. And what we see, again, we have the different methods on the bottom, is something very interesting. We have yellow as the FreeSurfer stats, and red is all of these bits of information together. And what we find is that we achieve the best performance when using information from all of these sources together. And specifically, we find performance that is equivalent to literature using the same sample and the functional data. So we see similar performance of other researchers using functional data of a task specifically designed to model aspects of working memory, which is very impressive that we can do it with the structural data alone. The other thing we see is that all of the regions from the PCA models and the deep neural networks perform significantly better than that of the free surface statistics. So if we include, again, information from the brain, either using deep learning or this sort of traditional method of PCA, we get improved performance. We've answered again that question of, is there information we're missing? Yes. Does it help us predict our outcome, both regionally and globally? The autoencoder whole brain does not work as well, however. It is still significantly predictive, but less so than any of the other methods. And I think if I had theories, this, again, taking the entire brain, that three-dimensional structure, learning these very complex low-level representations, it's a lot of data to learn patterns in concurrently. It's very difficult tasks. And I think this is really interesting because within a lot of the deep learning literature, this is the most common strategy. Take the entire brain, dump it into a model, and then use that information for prediction. But we see that we can improve our performance when we incorporate information that we know. PCA, however, actually does very well when we use a single PCA model. This is very sort of surprising to me, taking again the entire brain, flattening it, putting it into a single PCA model, and then using that information to predict our outcome. It does better than the free surface stats. And this piece of PCA performing so well was very interesting to me. So we're going to take a quick detour before we jump into working memory or externalizing psychopathology to talk about maybe why PCA is doing so well. Just some theories and supported with data, right? That's how we do this. And so here we have dots, and each dot represents a feature from each of those methods of getting information from those images. We have little dots that represent the PCA features that we've learned, the autoencoder features we've learned, and then these aspects of the extracted bits of information. The size of each dot indicates how important that feature was in predicting working memory. One model for each feature set. No relationship between these columns of dots right now. One of the issues with something like PCA, or even more significantly within the autoencoder, is that when we apply these transformations to this data, we lose some of that interpretability because we're learning these very complex representations of morphology. So with the free surface stats, I can point to features and say, these numbers are shape. They are thickness, volume, surface area. And these numbers are intensity, which we don't really have the same way with PCA or the autoencoder. And so we notice two things about this structure, whatever it is. One, shape, very important in predicting working memory. And it turns out that this structure is the middle temporal gyrus, the top performing structure of our modeling strategy. And we can look to see how correlated the features that we've learned from PCA and the autoencoder are to these representations of our data that we know. So we can look to see, indicated by sort of thickness of the line or color of the line, how correlated each feature from the deep neural network or PCA is to these higher level properties of the image. And what we see is that the autoencoder is focusing very heavily on intensity. But we know from the free surface stats that intensity is less predictive of working memory. And this might not mean anything except for in this structure, we see significantly worse performance with the deep neural network. So we have this question of, are we looking in the wrong place? And how is the charger? That's fine. I've got enough. So it's not wrong, right? Because the goal of PCA was, or no, autoencoder was reconstruction. Our goal was to compress and reconstruct. We weren't considering our objective at that time. So it learned what it felt was most important. And it learned aspects of intensity very heavily. It just so happens that those features are less predictive of working memory within this structure. This is not the case for the precentral gyrus, of which we see equivalent performance between PCA and the autoencoder. And we see, one, shape less important in predicting working memory, and two, the autoencoder, the features it's learning, represent sort of intensity and shape equivalently. It's looking at both places in terms of reconstruction. So one aspect that might indicate why, in this case, PCA is doing a little bit better, but my other theory that we're going to get into, I do like this detective piece, is this aspect of multicollinearity. And it sounds confusing, but it's not. It just means that the features that I'm using are related to each other. And we know that some of those high-level features are going to be related to each other. Volume and surface area are going to be very similar. And this aspect of multicollinearity creates issues both from a modeling perspective, leading to these sort of more unstable coefficient estimates, and from a piece of just information redundancy. If we have the same sets of features, we're just not including more unique aspects of the image. So what we can do with these two structures is look to see how correlated are the features from the deep neural networks of these two structures, one in the case where it's doing worse and one in the case where it's doing equivalent. And what we find is that in the model where we're getting lower performance with deep learning, we have significantly higher correlation among the features that we've learned than the precentral gyrus, which is performing equivalent to PCA. Now, what you will also notice is that I did not show the correlation among the PCA components because there are none. It's sort of hard to tell, but so each row is how correlated is this feature to that feature, and green is zero correlation. There's no relationship among those features. And PCA doesn't have any because it has this nifty little mathematical constraint, bringing the word nifty back, and that forces these features to be uncorrelated, which is really cool. This is not the case of the autoencoder. We have a lot of relationships among the features that we're learning, both high positive correlations and high negative correlation. So we've created issues both for the modeling, the subsequent modeling that we're doing, and just from a redundancy piece. We have more redundant information within the autoencoder. So again, two cases for why PCA might be doing so well. So let's summarize the results of working memory. Does information from structural MRI predict working memory? Yes. Can we do this better than the existing literature? Yes. Is it because we included new bits of information? Yes. Was it achieved using deep learning? Yes. Was it consistently deep learning that performed the best? No, it was not. Because again, we talked about reasons why PCA might be doing so well. But again, we achieved the best performance when using more information from the images. So now we get into predicting externalizing psychopathology. And this is where things get a little hairier. One of the things that I really want to reiterate is that we're not learning new information from the images. We've already built these models, both the autoencoder and PCA. We have these features that represent morphology and intensity. We're not retraining those models. We're just using those, that was cool. We're using these aspects from the data to just with different tasks, both predict working memory and externalizing psychopathology. We're just using them in different ways for different predictive tasks. So that's just, I don't know, wanted to be clear about that. And again, we know some of these aspects of the free surfer stats do predict externalizing psychopathology, albeit much less well. We see that the scale of the color bar is reduced and we see a lot less red, even within the free surfer statistics. It is a lot harder to predict psychopathology. We knew this, we talked about this, why it would be so hard. We'll cover it again. And what we also see is PCA is not doing as well as it was in the case of predicting working memory. We see even maybe some less structures that are predictive. We see a couple subcortical structures in the hippocampus, and but just maybe not as well, maybe not as many as in the case of the methods we used earlier. The autoencoder also maybe a little more orange. Again, hard to tell from these plots. But what we do see is when we include information from all of these sources together, now we start seeing red, which sounds scary. But when we look at the top 3 predictive structures, what we find is that the best performance we obtain is from using information from all of these feature sets together. So using information from the free surfer stats or the autoencoder, the deep neural network, or PCA, using information from all of these sources gives us the highest performance regionally. And I'm sounding like a little lawyery, but that is because when we look at this aggregate prediction, what we find ultimately is that the best performance that we could obtain in predicting externalizing psychopathology came from using the high-level statistics from the left hemisphere specifically, which is really, really interesting. And maybe, maybe not always the result we want, but the result that we get. We knew that externalizing would be a lot harder to predict. Whole brain, not better than random chance. Again, very hard to predict psychopathology. We can do it, but just not better using features that we obtain from deep learning and PCA. So what do we walk away with? Does information from structural MRI predict externalizing psychopathology? Yes. Can we do it better than the existing literature? Yes. Is it because we included new information? Ultimately, no. We did see some improvements when including additional information regionally within specific structures, which inherently is interesting, but ultimately the best performance in looking at the brain in the context of mental health came from using these high-level properties of that data. And so why might this be? Well, we knew it would be very hard to predict externalizing psychopathology. Again, measurements comprised of these subjective symptoms, and the measurement that we're using to assess this is endorsed by the parent. So there's a lot of sort of abstraction happening there. This sample is not enriched for psychopathology. It's trying to encapsulate the sort of a normative population. It is not, for those that have more severe forms of externalizing psychopathology, we have less data available, which makes it a little more hard to model in that region. And lastly, again, I'm going to say it over and over, we're tying these very complex mental and behavioral symptoms back to these structural properties of these complex neural pathways. It's a very difficult task. One of the things that I also want to sort of put out there is, why would the inclusion of low-level information help us predict working memory, but not externalizing psychopathology? So why would additional information sort of help in one region, in one task, and not in the other? If we know that our data X is weakly associated with our outcome, then adding more detail about that outcome might just provide just unnecessary granularity. Maybe it is those high-level properties that sort of get the most robust information out. That feature space is also a lot smaller. When we explode the number of features, it becomes more and more complex to model these data. Also, we have this risk. When we go back to the raw imaging data and we get information directly from that source, we're at risk of introducing some of the noise that existed in the image in the form of dropout or motion back into the data, something that still exists to some extent within those high-level properties, but less so than when we're looking at the imaging data directly. So looking forward, why am I still somewhat optimistic about deep learning? There are infinite architectures. There are, again, we talked about that tuning purgatory. There are millions and infinite ways, which is more than a million, to structure these neural networks. We could evaluate a couple of them. They took an exorbitant amount of time to train, a very long time, so we can only evaluate several of these networks. We also have this sort of rapidly expanding body of research. Deep learning is advancing, and with it, we have rapidly advancing hardware. As the hardware gets better, we can train more models, we can train more models faster, we can try more combinations and get more unique structures. This was probably the biggest limitation within this entire analysis, was how long it took to deal with the data, to train the data, to process the data, to feed it through the network, only to say it's not working and do that over and over. But again, limited by the hardware, which is improving. And then lastly, having access to these larger neuroimaging data sets will hopefully help us keep learning more complex patterns. In some of the most successful computer vision tasks within deep learning, they are trained using millions and millions of images. This is not what we had, and our images were very complicated. So including additional information might help with some of those supervised models. It's hard to say, but we can only benefit with including more data. Again, we need that hardware to speed up with it though. And so with that, I am going to thank so many people, everybody in this room, everybody that I worked with, all of my advisors, everybody. It's one of those things where you say, like, I know a lot of people helped me along the way, but I could not have done it without my advisors, my cohort mates, my friends at the gym, just everybody within there. So with that, I want to thank everybody, and that's it. Whoa. Thank you for the claps. Okay. Yes.
Or you can repeat everyone's question.
I don't want to run around. I'm pretty sweaty. Okay. Yeah. Questions from anybody about anything related to the project or school. Nate, I see you. I see you over there.
So I'm kind of curious to see if you said that the information from all three algorithms, the autofocus, the three stats, and the PCA perform best. Did you compare that to information from, for instance, 3 different PCAs or 3 different autoencoders? Getting at the idea that it could be advantages from like an ensemble effect than from individual algorithms.
Yeah, so the question is about training sort of multiple instances of the neural networks and the PCAs. Yeah, over different folds of training. So this gets at to like, are you talking about uncertainty quantification?
Or just that like ensembles that have shown to work well because they produce finance volumes and contain variance in that fast and straight up.
Yeah. So we did the final performance. So again, with limited time, it's hard to jump into all of the aspects, but all of the performance that we showed was on test data, a global holdout test. So data that the training never saw. And it was global, meaning that we have different folds of the training data. So with that, we create 10 PCA models for each of these splits. So when we talk about like why it took so long, we're training 10 instances of these models every time. And with the neural networks, there's a lot of stochasticity. Are there like a little bit different every time? And so that can help reduce sort of the variability in the predictions. And yeah, it did help globally. But the results that you've seen are from those sort of averaged, the results that we're seeing are from the 10 splits, the box plot, not ensembles of splits, if that makes sense.
I have a question online. For your neural networks, CNN or AE, did you use random weight initializations or another initialization?
Yeah, that's a good question. So talking to you about how these networks learn those weights and those kernels, you can either just make them random or use different methods. And we tried a lot of the different strategies that the Python library PyTorch has without really any difference in performance, which was kind of interesting. Nate, go for it, yeah.
If you had another 6 months or a year, are there any other architectures you would want to try?
We're thinking about graph neural networks still. I do like the idea a lot. I think if anything, if I had more time, I would go back to the functional data. Even though it's noisy, even though, it's very noisy. I know, I see my advisor's face, but the autoencoders worked very well, and we didn't use the autoencoders with the functional data. So I think there are ways that we could get about getting that sort of those latent features from the functional data that might be interesting. We could even use generate, I'm getting it, see, this is, getting ahead of myself, but I would go back to the functional data. Kayleen, and then
I know you kind of talked a little bit in the background, but how advanced is like the integration of machine learning into neuroscience, whether it's like MRI or EEG or anything?
Yeah, so a lot of the advancements within deep learning are comparatively relatively recent. And again, with that hardware getting better and better, it's going to keep improving. Even like tangentially using tools like ChatGPT for boilerplate code or like from those aspects. But in terms of modeling, like even, smaller of like wet lab aspects of like at the cellular level, you can build these complex simulation-based models in ways, and again, with that sort of flexible configuration that you couldn't do with a lot of other strategies. So I think we will see it more and more. I would say I went into this as like an AI skeptic, but even though we ran into a lot of roadblocks, I still feel optimistic about the field and the utility of the tool. Lisa.
So do you think if you had kind kind of like a balanced cohort design or something, that the performance would have been different, like if you kind of had more of like a case control type of design?
Yeah. No, yeah. So that was that piece that we talked about right away of like looking at the dimensions of psychopathology and not sort of binary diagnoses. I did actually look at binarizing the externalizing the values are these like T-scores, and there is a clinical threshold of 63. So I did binarize that data by the clinical threshold and then try and just build classifiers. It did not work, but again, very unbalanced. We had a lot more, healthy controls than we did those with externalizing psychopathology. So yes, from some earlier work we did in predicting suicidality, balancing the data set was like one of the more critical aspects of prediction. Yeah.
So, was this all done in volume space, and did you try to learn the projections like into the
It was done in volume space. So one of the ideas that would be really interesting would be to use these. So rather than using these like volumes where the brain, every pixel represents spatially some region, we could build these like mesh surfaces in what is surface space, and then apply convolutions over that mesh surface, which is confusing, but yeah, I thought about that, but I didn't see I saw the bulk of the research using structural MRI within the volume space. So I went with that. But I think it would be cool to go into the surface space, which sounds fun.
Got another question online. It says, I'm curious whether you explored transfer learning or techniques like multitask learning. Were these ever used or candidates for exploration?
Yeah, so that was, can I ask who's asking? Is it okay to ask who's asking?
Both of those questions were from Daniel.
Daniel, yeah. Great question, Daniel. Yes, that was sort of the initial goal of the entire project, was to build these networks in sort of what's called cumulative learning, or no, curriculum learning, where we build these networks to predict this easier task like working memory or inhibitory control, something we can do better, and then use those networks as the starting point for subsequent networks in predicting externalizing psychopathology, i.e. transfer learning. But the supervised models just, at best, they would work equal to extracting features in these unsupervised networks. So transfer learning did not help. But good question.
Any other questions? He's got funny questions. He was ready to come, so we'll also let you have that question. Still have